Our next speaker is a master student from Dr. Hugh Kim. It's Stephen, the role of Filamin A, signaling platelet thromboxane A2 secretion. There you go.
Hi everyone, I'm Stephen, and in this presentation I'm going to talk about the role of Filamin A in thromboxane A2 secretion. So here's the outline. I'm going to talk about, so let's go over the background first. What are platelets? Platelets are responsible for hemostasis. They're responsible for the formation of blood clots. And their action depends they really depend on the secretion of this compound, thromboxane A2. So it's an autocrine activator of platelets. Upon platelet activation, this compound is secreted, which feeds back, allowing more platelet activation. Platelets depend on thromboxane A2 secretion to function. If you inhibit its secretion using aspirin, platelets don't clot properly. But we do not understand the pathway the intracellular signaling pathway that leads to the generation of thromboxane A2. And my lab thinks that this protein called Filamin A plays a role in thromboxane A2 generation. So Filamin A, it's an actin-binding protein with intracellular signaling protein binding roles. And the function of platelets depend on this protein. If you knock this protein out of mice, they have a prolonged bleeding time. And if you isolate these platelets from these mice, they don't clot properly either. So that's very interesting. My lab generated a conditional knockout of Filamin A inside platelets. So in knockout mice shown right here, there's no Filamin A in platelets, but there's Filamin A in all other tissues. In control mice, there's Filamin A everywhere. So my hypothesis is that Filamin A contributes to platelet thromboxane A2 secretion. This study was done by Dr. Kaliangola. He showed that if you knock Filamin A out of platelets, they don't secrete a lot of thromboxane A2 upon stimulation. And from that finding, I want to explore the mechanism behind this defect. So I started looking at a group of kinases called MAP kinases. Just to recap, MAP kinases, they are ubiquitous in most eukaryotic organisms. In platelets specifically, upon thrombin stimulation, MAP3K, MAP2K, and MAP kinases, they are sequentially phosphorylated and activated. Which the three MAP kinases that are important, they are P38, ERK1, and ERK2. These three MAP kinases cause the phosphorylation and activation of cytosolic phospholipase A2, which is directly responsible for the generation of thromboxane A2. My hypothesis is that in Filamin A knockout platelets, the phosphorylation and activation of these MAP kinases and cytosolic phospholipase A2, they are defective. And that's exactly what I have found. This is the densitometry result of these Western blots. Here in black, they are the control platelets. Upon thrombin stimulation, there's lots of ERK1 and 2 being phosphorylated, a lot of P38 being phosphorylated, a slight but above baseline level of CPLA2 being phosphorylated. In knockout platelets shown in white, none of these is observed. So here's a summary. Without Filamin A, none of these proteins I have just talked about, they are phosphorylated and activated. And that's why thromboxane A2 is not secreted inside these platelets. Again, I want to figure out the mechanism behind that. So my hypothesis is that Filamin A associates with these proteins right here. And that's why if you knock Filamin A out of platelets, they are not activated. So I did a co-immunoprecipitation assay. I pulled down Filamin A and all of its associated intracellular signaling proteins. So in all cases, filamin A is pulled down, but none of the signaling components, they are not pulled down. So from this, we can see that filamin A does not associate with any of the intracellular signaling proteins. That's all of the experiments I have done, so let's conclude this presentation. So just to recap, filamin A binds to many intracellular signaling proteins. This has been shown in platelets and in other hematopoietic cells. Filamin A, very important for thromboxane A2 secretion. It is also responsible for the phosphorylation and activation of ERK1 and 2, P38, and CPLA2. But it does not directly associate with any of them. The next step of my project is to figure out whether filamin A contributes to the activation of MAP2Ks, which are one step above the activation of MAPKs. With that, I would like to thank my PI, Dr. Hugh Kim, and all of my lab members for helping me. I would also like to thank the funding agencies shown right here for making this research possible. Thank you so much.
Thank you very much, Stephen. Any question for Stephen?
So the filamin A is distributed throughout the platelet. There's a good population of it, I think, on the platelet membrane skeleton. Do you think that there's a differential role for the different populations in this? It would seem like it would be the ones that are next to the skeleton.
Sorry, can you repeat your question again?
Well, at least back in the day, one would say that that the platelet cytoskeleton is composed of two components. One is the piece that's immediately under the membrane, the platelet membrane skeleton, and then there's the one that has the core of the actin deeper in the platelet. And those two, when a platelet becomes activated, get cross-linked. And filamin A is at least, I think in both of those populations, but I know that it's at least in the membrane skeleton because it's attached to platelet GP1B alpha. Do you have any information as to whether those populations are differentially involved in activation that leads to production of thromboxane A2?
I do not have any data to show here, but it's just a speculation. From an earlier presentation presented by Felix, my coworker, he has found that protein kinase C signaling pathway is hampered in filamin A knockout platelets. And protein kinase C, if they're activated, they are closer to the plasma membrane than the inside of the platelets. So I would speculate that filamin A, it should be at the cortical of the platelets that are more involved in this pathway, but that would warrant further studies to study that.
Thanks.
Any more question for Stephen?